Welcome back to Mechanical Engineering Made Simple. Today we are going to talk about something that sounds, well, incredibly mundane on the surface, but is actually responsible for the difference between a life-saving medicine and a vat of toxic sludge. We are talking about mixing.
It is deceptive, isn't it? When most people think of mixing, they think of, a KitchenAid mixer or stirring milk into coffee. It feels intuitive.
Right.
But in the pharmaceutical industry, mixing is often the hidden killer.
Yeah.
It is the silent variable that can ruin years of research and millions of dollars.
Exactly. And we're not just talking about stirring things faster. We're diving deep into the engineering reality of how drugs are actually made, based on the work of Paul, Midler, and Son. The stakes here are just incredibly high. It turns out the difference between a successful drug and a total failure can come down to the geometry of a turbine blade.
That's right. And to understand why it's so high stakes, we have to look at the mission of these facilities. If you look at petrochemicals, oil and gas, they build these massive plants designed to do one thing continuously, 24/7.
Right, they're optimized for a single steady state.
Exactly. You turn it on and it runs for years. But pharma is different. They use what are called multi-purpose batch reactors.
Which sounds efficient from a business perspective, one factory, many products. But I assume that creates a massive engineering headache. You're asking one machine to be good at everything.
Precisely. You have this one vessel, this one tank, and on Monday it needs to heat a reaction to boiling. On Tuesday it needs to distill a solvent. On Wednesday it needs to crystallize a solid product. It has to be a jack of all trades. And making one machine do all of that without ruining the delicate chemistry inside, that is the core challenge.
So let's start with the equipment itself. When I picture a chemical reactor, I just picture a big stainless steel tank, standard stuff. But according to the source material, that's not always the case in pharma. In fact, steel is often the enemy.
No, often it can't be steel.
Yeah.
In fine chemicals, you're frequently dealing with highly corrosive acids.
Like hot hydrochloric acid.
Exactly. That would eat right through 316 stainless steel in a matter of days. You'd have leaks, contamination, structural failure. So the industry standard is the glass-lined vessel.
Which sounds incredibly fragile for an industrial environment. I mean, we're talking about thousands of liters of liquid swirling around.
It's surprisingly robust chemically, but it introduces this massive mechanical paradox. These vessels can range from 80 liters up to 20,000 liters.
20,000 liters?
Imagine a 20,000-liter glass bottle encased in steel. The problem is, as an engineer, how do you fabricate the internals? You cannot weld glass.
Right. If you have a standard steel tank, you can weld baffles to the wall, you can weld complex, sharp impeller shapes to the shaft, but with glass-lined steel, you're stuck. You can't just spark up a TIG torch.
You are severely limited. The source material explains that because you have to coat the metal agitator in glass, you can't have sharp edges. Everything has to be rounded.
Why is that?
If you have a sharp 90-degree angle, the glass coating will crack due to surface tension during the firing process. So traditional pharmaceutical mixers, what we call retreat blade impellers, have these soft, rounded geometries.
Which changes the fluid dynamics completely. It's like trying to row a boat with a spoon instead of a paddle. You don't get that same bite. But there's a detail in this text about assembly that I found fascinating. It's how they actually get the blades into the tank.
Ah, yes, the cryolock and Elkalock systems. This is a brilliant workaround for the small hole, big part problem.
Right, because the tank has a small manway at the top, but the impeller needs to be big to be effective, too big to fit through the hole. In a metal tank, you just bolt the blades on inside. But you can't do that with glass, can you?
Exactly. Bolting creates stress points and stress cracks glass. So manufacturers develop this cryogenic shrink-fitting technology. They make the drive shaft and the impeller as two separate glass-lined pieces. They lower the shaft into the tank. Then they pump liquid nitrogen inside the hollow core of that shaft.
So they're freezing the metal from the inside out.
Yes. The extreme cold causes the metal inside the shaft to contract. It shrinks just enough, we're talking millimeters.
Okay.
Then, while it's shrunken, they slide the impeller hub over the shaft. They stop the nitrogen flow, the shaft warms up, expands, and it creates this incredibly tight, seamless interference fit.
That is just cool. You're using thermal contraction to assemble a glass machine inside a sealed tank. No bolts, no gaskets.
It's brilliant engineering, but it highlights the constraints. And there's another massive trade-off with glass. Glass is a ceramic. It's a terrible conductor of heat.
Which is bad news if you have a reaction that produces a lot of heat, right? You
That's
need to get
a
that
nightmare
tightly.
for process control. The source notes the heat transfer coefficient is significantly lower than steel. Plus, you have a thermal shock limit. The maximum temperature difference you can have between the coolant jacket on the outside and the liquid inside is only about 125 degrees Celsius.
Wait, so if the reactor gets too hot, you can't just blast the jacket with ice-cold fluid to cool it down.
If you do, you shatter the glass lining. It's like pouring boiling water into a cold glass cup.
So you have to be gentle.
Very gentle. This forces you to run highly exothermic reactions, much slower than you otherwise would, just to manage the heat. You are throttled by the glass.
So you have rounded blades that don't mix aggressively, and a tank that acts like an insulated thermos when you actually want to cool things down. That sets the stage for what I want to talk about next, the actual chemistry. Because if the mixing is compromised, the chemistry starts to do strange things.
Let's get into the kinetics. This is where it gets really interesting for anyone interested in why scale-up is so hard.
The source material calls this mixing-sensitive reactions. Now, to a layperson, mixing liquids sounds easy. I pour milk into my coffee, it mixes. Done. Why is this hard?
Because in your coffee cup, you don't have a chemical war going on. In pharma, you have to distinguish between macromixing, which is just moving the bulk fluid around, and micromixing, which happens at the molecular scale.
And the problem arises when the mixing speed is slower than the chemical reaction speed, correct?
Exactly. This is the concept of competing timescales. Let's look at the competitive consecutive reaction scenario described in the text. This is a classic trap. Imagine you have reagent A in the tank, and you're adding reagent B. You want A plus B to react and form product R. That's your drug.
Okay, A plus B makes R simple enough.
But there's a catch. Product R is also reactive. If there's a lot of reagent B hanging around, product R will react with it to form substance S. And substance S is waste. It's junk.
So it's a race. You want A and B to find each other, but you need to stop B from then attacking the product you just made.
Correct. Ideally, you want B to be consumed by A instantly.
Yeah.
But if your mixing is slow, when you pour B into the tank, it doesn't disperse instantly. It creates a hot spot, a localized cloud near the feed pipe where the concentration of B is extremely high.
And inside that cloud, B eats up all the local A, makes R, and then, because there's still tons of B left, it immediately eats the R to make S.
Precisely. You end up destroying your product the moment you make it, simply because you didn't spread the reagent B out fast enough.
And the source points out this is the classic scale-up trap. In the lab, in a 1-liter beaker, you can mix things almost instantly.
Right. But then you move to a 10,000-liter tank. The circulation time, the time it takes for fluid to loop around the tank might be 20 or 30 seconds. In chemical terms, that's an eternity. The damage is already done.
And we aren't talking about losing just a little bit of product. We are talking about the 0.1% rule.
This is the regulatory hammer. In the pharmaceutical world, if an impurity level rises above 0.1%, you often have to register that impurity as a new chemical entity with regulators.
Which means toxicity studies, paperwork, massive delays.
It's not just paperwork, though. The source mentions these impurities can physically wreck the process downstream.
Ah.
A tiny amount of byproduct can change the surface tension, causing it to foam uncontrollably. Or it can turn into a gel that makes filtration impossible. You might have a tank full of product, but if you can't filter it, you have to dump the whole batch.
So 0.1% is the difference between shipping the drug and shutting down the plant.
It absolutely is.
Let's ground this in a real-world story. The text breaks down the gas wars involved in synthesizing an antibiotic called cefoxitin. This story really shows what happens when mechanical mixing just fails.
This is a fascinating case study because it shows a war on 3 fronts: chemistry, mechanics, and regulation. So in making cefoxitin, there's a step where they react 2 intermediates. But this reaction produces hydrochloric acid HCl as a byproduct.
And as we mentioned, HCl is nasty. But here, it wasn't just corrosion, right?
No, here it was acting as a catalyst for decomposition. In this specific reaction, if the concentration of HCl hits just 0.1 molar, it destroys the product.
Okay, so the obvious solution is to remove the HCl.
You can't remove it all. If you remove it completely, the reaction stops. It needs a tiny amount of acid to proceed. The source says the Goldilocks zone was exactly 0.004 molar.
That's an incredibly tight window.
It is. So the engineers first tried mechanical solutions. They tried distillation, boiling off the HCl gas as it formed.
Too slow.
Way too slow. Mass transfer from liquid to gas is limited by surface area. By the time they boiled it off, the concentration in the liquid had already spiked and killed the product.
Oh, what did they do?
They turned to molecular sieves. These are solid materials with tiny pores that trap specific molecules. They act like a sponge for acid.
But the source makes a point of the physical form of these sieves. They couldn't just use standard pellets.
No, pellets were too slow. The diffusion of the acid into the center of a pellet took too long. It's the same problem again, mass transfer limitations. So they had to use powdered sieves, 1 to 4 micrometers in size, to get more surface area.
But even that wasn't enough, was it?
No, it wasn't. They hit the absolute limit of what physical mass transfer could do. Mechanical mixing and physical adsorption were just too slow. So they had to introduce a homogeneous scavenger.
A homogeneous scavenger? What's that?
They added a chemical trimethyl carbamate that reacts instantly with HCl in the solution.
So they essentially hired a chemical mercenary to neutralize the acid the microsecond it was formed.
Exactly. It bypassed the mixing problem entirely. Because the scavenger was dissolved in the liquid, it was right there. No mixing required.
And the takeaway there is crucial. Sometimes you cannot mix your way out of a problem. You have to change the chemistry to survive the equipment.
That is the lesson. Don't bang your head against the wall trying to design a better impeller if the physics of diffusion won't let you win. Change the game.
Speaking of limitations, let's move to another section. The expert calls this the most difficult operation to scale up. We're moving from liquids to solids. Crystallization.
Crystallization is a nightmare because the requirements are completely contradictory. To mix chemicals, as we just discussed, you usually want high energy. You want turbulence.
Right, smash it all together. High turbulence, good mixing.
But to grow a crystal, you need gentleness. A crystal is an ordered lattice. It takes time. If you hit a growing crystal with a high-speed blade, you shatter it. You break the lattice.
It's like trying to build a house of cards inside a tornado.
That is a perfect analogy. And the source gives a great example involving a side chain for an antibiotic. It worked fine in the lab, nice big crystals, and they moved to the pilot plant and disaster struck.
They got what the text calls a bimodal distribution.
Which is engineer speak for we have some giant chunks and a massive amount of dust. They call the dust fines.
And fines are the enemy of manufacturing. Why?
Fines clog the filters. Instead of liquid flowing through a pile of rocks, it's trying to flow through mud. It can take days to filter a single batch. Plus, fines trap impurities. The process just ground to a halt.
So they investigated what went wrong.
It turned out to be a double failure. First, the reaction was happening too fast. It was causing supersaturation to spike too quickly. High supersaturation triggers nucleation, which means instead of growing existing crystals, the liquid just spawns millions of tiny new ones.
So that creates the dust.
Yes.
Yeah.
And on top of that, the impeller they were using was spinning too fast, smashing the crystals that did manage to grow.
So they had to slow everything down.
Drastically. They extended the time they spent adding reagents from 2 hours to 6 hours. This kept the supersaturation low, allowing the crystals to grow slowly.
But they also changed the hardware. This is where we talk about the Ocado intermig.
Yes, the intermig is a specialized impeller. Unlike a standard turbine that pushes everything out with high force, the intermig consists of multiple stages that create a chaotic up and down flow. It provides excellent circulation keeping solids floating, but with very low shear. It doesn't act like a blender.
It gently lifts rather than chops.
Exactly. And this leads to a critical lesson about scaling up. There's a standard rule called equal power per unit volume. It says if you have a big tank, you should put in the same watts per liter as you did in the small tank.
That sounds logical. Preserve the energy density.
For mixing liquids, it usually works. For crystallization, it is dangerous. Because as you scale up the tank, if you keep power per volume constant, the tip speed of your mixer blade increases.
The tip speed being how fast the outer edge of the blade is moving.
Right, and tip speed is what breaks crystals. It's the impact velocity. So in this case, if they had followed the textbook rule, they would have kept destroying their product. They actually had to use less power per volume to save it.
This brings us to the final dilemma, the jack-of-all-trades problem. We mentioned at the start that these plants use multi-purpose reactors.
This is the economic reality. You have one vessel. On Monday, you might be doing a hydrogenation.
Which involves gas.
Right, hydrogen gas. To get gas to react with liquid, you need to chop the bubbles up into tiny spheres. You need high shear. You need to spin that impeller fast, chopping violently.
But then Tuesday comes around and you're doing that crystallization we just talked about.
Where high shear destroys the product. So you have the same motor and the same shaft needing to do two physically opposite things.
This is why the source emphasizes variable speed drives, or VSDs.
It sounds simple, just a knob to turn the speed down. But historically, industrial motors were often fixed speed. Adding a VSD is capital intensive. But without it, you're dead in the water. You can't just pick one speed.
And there's a legal trap here, too, called the biobatch.
This is crucial. When you're getting a drug approved by the FDA, you run a specific production batch called the biobatch. This is the batch used for the pivotal clinical trials.
The one that proves the drug actually works in humans.
Right. Once that biobatch is approved, the FDA essentially says, okay, you can sell this drug, but you have to make it exactly the same way. You are legally locked into those physical attributes.
So if your biobatch had a certain mixing profile, you have to replicate that at full scale forever.
Yes. If you optimized your biobatch using a specific mixing speed that gave you a certain crystal size, you must be able to hit that target in the 20,000-liter tank.
Yeah.
If your big tank can't match the biobatch parameters, you don't have a product. You have a regulatory failure.
That's terrifying. You aren't just designing for physics, you are designing for compliance. The constraints of the vessel define the legality of the drug.
It connects back to the very beginning. The mixer is not just a stirrer. It is the device that determines purity, yield, and legal compliance. It is the heart of the process.
So let's unpack this all. We've learned that mixing is this incredible balancing act. You're fighting corrosion with glass, which limits your shape and heat transfer. You're fighting chemistry that wants to eat your product, so you need to mix fast. But you're fighting crystal breakage, so you need to mix slow.
And you are doing it all in a giant tank where you can't even see what is happening. You rely entirely on calculations and sampling.
It gives you a new appreciation for that little pill in the medicine cabinet.
It certainly does.
Yeah.
And it raises a provocative question. When we look at the history of drug development, we see many drugs that failed in phase 3 trials or were canceled because they couldn't be manufactured reliably.
Right. The unscalable drugs.
We have to wonder, how many life-saving drugs failed not because the chemistry was wrong, not because the molecule wouldn't have cured the disease, but simply because we couldn't build a mixer that worked at 20,000 liters? How many cures are lost in that scale-up gap?
That is a heavy thought to end on. It really shows that mechanical engineering and chemical engineering are two sides of the same coin. You can't have the cure without the tank.
Absolutely.
Well, on that note, we are going to wrap up this deep dive. Thank you for listening. And if you haven't already, get over to our YouTube channel, Mechanical Engineering Made Simple, where we post hands-on how-to videos, visual breakdowns, and practical engineering walkthroughs you can actually use. A family that solves together stays together.